Hello everyone, welcome to our final Immunolab lecture, and also our final lecture in the immunology series. So I hope you guys have enjoyed it so far, but today we're going to be looking at antibody-based methods of immunology, and we'll be focusing on immunofluorescence and flow cytometry. So in this picture we have immunofluorescence. So we have IMF staining of influenza virus infected lung tissue. So these cells were treated with fluorescent dye, tagged antibodies against a specific virus. So the nucleus of the infected cells is bright yellow-green. So here we can see influenza virus in the lung. And this is indicating the location of the viral antigen. So again, these can be used for many diagnostic things and as well as studying various different cells. So immunofluorescence. So antibodies can be used to detect specific molecules expressed on the cell surfaces in tissues, in biological fluids, and many other types of samples. We're essentially adding a tag to an antibody that allows ready detection of binding to the specific target molecule. Fluorescent dyes are used for imaging by immunofluorescent microscopy. So we use something called a fluorescent, which is one of the several fluorescent dyes that can be complex with antibodies. So there's several different types of these and a bunch of different names for different colors and etc. But essentially, we have an antibody molecule that has a fluorescent dye molecule attached. And then we have this antibody. Then our antibody will bind to the original antibody, causing that antibody I discussed. But this is referred to differently in some textbooks. So, like I said, there's many different names and excitation and emission frequencies with different colors: orange, green, red, orange, etc. So the dyes used for IMF are excited by light of one wavelength and emit light of a different wavelength. So the emitted light is viewed using a fluorescent microscope or flow cytometer. The excitation and emission wavelengths for several fluorochromes, as are seen up here. Commonly used dyes are fluorescent isothiocyanate or FIT-C and phycoerythrin or PE. These dyes can be conjugated to antibodies. Light emission from dye conjugated antibodies allows for detection of the protein the antibody is binding to by immunofluorescent microscopy. So again, we shine with one wavelength, so our exciting light. Then we have our emitted light, which is a different wavelength. So we shine here, and then we get a green emission in this case. So the green triangles represent the fluorescent dye FIT-C. Exposure to light at the excitation wavelength causes FIT-C to emit a green light. This is detected by fluorescent microscopy or by flow cytometer. This example shows direct IFM, which is the fluorescent dye is directly conjugated to the primary antibody. So here we don't have that second antibody like we've seen before. That's not there. So this is an example of a primary antibody. The antibody binding to the antigen, the protein of interest. So again, whatever our interest antigen may be. So an example of its application. So here's an image of pancreatic islets of Langerhans labeled with anti-GAD and anti-glucagon antibodies tagged with different fluorochromes. So the beta cells produce GAD and alpha cells produce glucagon. So the use of different antibodies conjugated to different fluorescent dyes allows visualization of different cell types in the same sample. So again, we can tag a bunch of different stuff and have a kind of visualization. So we see almost the red within the green, so it allows us to give that crisp image. So in this example, antibodies specific for glutamic acid decarboxylase, or GAD, were conjugated to FIT-C. Antibodies specific for glucagon were conjugated to PE. And exposure to light of the excitation wavelength, the FIT-C becomes a green light and PE becomes orange light. The beta cells appear green and the alpha cells appear orange. So you got that bright orange, and green in the inside. So direct immunofluorescence. Oops. Sorry about that. So now we'll take a look at direct immunofluorescence. So antibodies specific for the antigen of interest are directly tagged with immunofluorescence. So this was that primary one we saw. So again, there's no that secondary antibody, so that's not there. So we have an incubation with the tagged antibody allows binding. A washing step removes the unbound antibody. Bound antibody can then be visualized by fluorescent microscopy or by flow cytometer. So here's that picture we saw at the start. Now we have indirect, so now we can actually see the antibody to the original antibody. So the antibody specific for antigen of interest is incubated with the target. The washing step removes the unbound antibody. Fluorescent tagged antibody specific for the first antibody are added. This binds to the first antibody during incubation step, and again, washing step removes the unbound antibody. The bound secondary antibody is visualized by fluorescent microscopy or by flow cytometry. This indicates binding of the primary antibody, and indirect IMF is more sensitive than direct IMF, so that's very important. So indirect is more sensitive, since more than one tagged secondary antibody may bind to the first antibody. So indirect IMF, the antibody to the antigen of interest, is not itself tagged. Instead, its presence is detected by a fluorescent tagged antibody or anti-antibody to the original antibody. So now we take a look at flow cytometry. So flow cytometry is used to measure cells labeled with fluorescent dye-tagged antibodies. Both the number of cells labeled and the intensity of the labeling can be measured, allowing determination of cell number or percentage, expressing a marker of interest and the level of expression of the marker. So these are used quite commonly now, and there's even certain degrees where you can become a cytometry technician. So if you're interested in some careers in immunology, that's an option. They're not insanely complex, but they're quite advanced, and there's a lot of advanced technology put into this. So you can see there's a lot of light physics, essentially. When we use our laser emitter, we have our mixed cell population, and essentially it's filtered through this extremely pretty powerful laser, and we have our detector. So as these pass through, these can essentially determine which cell it is based on the tag, the fluorescent tag, and they're separated into different containers through the machine. They mostly give results something like this, where they can provide, so red, here's our red, here's our green, and it's kind of a scale. So if something's in the corner, it's highly green. You know what I mean? So we can see charts. So essentially, what you'll see in these graphs is pooling, usually, of certain cells in a certain area. So we know that green's going to be all in one area, red in one area, and then cells that aren't either might be spread in between, and then the computer can count. So again, here's basically an infrared. You can do this also as well. It's not infrared, but you can see the colors. So here's all our neutrophils, monocytes, lymphocytes. Again, they're all tagged with different colors, so you can see the color distribution changes. So flow cytometry and fluorescent activated cell sorting, or FAC. So cells are labeled using different antibodies conjugated to different fluorochromes, like we said. So here we can see different fluorochromes. A single stream of cells is forced through the nozzle, passing through the laser beam, exciting the fluorochrome. So if you think about how tiny that is, so cells are shuffled through the laser beam one cell at a time. And then again, we have different wavelengths of light and stuff like that going through. So these fluorochromes are on bound antibodies which are attached to the cells, emit light. Like we said, we had that emitting light. These signals are detected by the photomultiplier tubes, or the PMT. You can see. Then we have scattered light, which is also detected, which is indicator of cell size. So the side scatter and forward scatter. So when this cell comes through the light, it obviously will disturb the light, and obviously the bigger something is, the more disruption it will cause, and then these will be detected by our two scatters, and then this will help detect the color with red, and then we also have our green photomultiplier. So flow cytometry allows for rapid and accurate characterization of many cells. The number of cells expressing certain cell surface molecules can be determined, as can the level expression of the cells. This is the fluorescent intensity. So this is used in a lot of conditions to diagnose certain rare diseases in certain types of cells. So again, cells are labeled by immunofluorescence, are passed through a nozzle to create flow of single cells. Here's our single cells going down. The cells pass through a laser beam, allowing determination of the various properties. So again, we have our cell size through the scatters, which influences the degree of forward angle light scatter, which are the falls, and then the larger cells scatter the light over a wider angle, and there'll be a high forward scatter. Cell granularity influences the degree of side scatter, side angle light scatter. So again, we had our side and forward scattered measurements, and cells with many granules produce more side scatter. So again, this is giving us so much more detail compared to ELISA, which is obviously a more simpler technology and much cheaper, where that's essentially more just counting or seeing if something is present. This can actually tell us about the cell itself. So we have absorption and scatter of laser light gives more information about cell properties, like I said. So cell size influences the degree of forward angle light scatter. Again, larger cells create a high forward scatter. Cell granularity affects the side scatter. So for example, neutrophils. Neutrophils are large and granule dense, so they have a high FALS and a 90-degree scatter. Monocytes are smaller and have few granules and have an intermediate level of FALS and 90-degree scatter, where lymphocytes are the lowest FALS and a 90-degree scatter. So again, here we can see we're on a scale, our 90-degree scatter and a forward scatter. With neutrophils being the densest and largest, they have the greatest forward scatter, like we see here, and then again we see intermediate and then low. So the amount of fluorescence due to the binding of labeled antibodies is measured. So this shows labeling of thymocytes with anti-CD4 tagged with FIT-C and anti-CD8 tagged with Texas red. So the double negative cells have no fluorescence in our lower left quadrant. So here. So if they were higher green, they'd be up here, more red up here, and then what's in between. So CD4 positive T cells are fluorescent with green and in the lower right quadrant. So you can see, these are always usually divided in quadrants, by the way. Then we have our CD8 T cells, which are fluorescent red in the upper left quadrant. So following our red fluorescence. Then our double positive cells, which again both have CD4 and CD8, versus our single negative cells here, single positive, sorry, and our double negative. These fluorescents are both red and green and are on the upper right. So again, they would form that. So that's quite messy, but I hope that's clear. The number and density of the dots reflects the number of cells in each category. So this is what's called the dual parameter plot, showing results obtained with two different antibodies labeled with two different fluorochromes for CD4 and CD8. And since there's cells that have both, which are double positive, they end up here. So again, CD8 up here, double negative, CD4, CD8, or double positive. So representation of data obtained by flow cytometry. So results from analysis of a sample of B cells expressing both IgM and IgD detected using anti-IgM, anti-IgD, conjugated to different fluorochromes. So we have histograms, which are useful to show the expression of one parameter, the size, either size, granularity, or expression of IgD or IgM. So then we have our two color plots, which you can see here, which are useful to display results with two or more parameters. So these plots show fluorescent intensity, indicating the expression of IgM on the x-axis and IgD on the y-axis. So here we see IgM and IgD. We have cells in the lower left part of these plots do not express IgM or IgD, similar to the other plot. So again, lower left, they're not either. Cells in the upper right part of each of these plots express the highest amounts of both IgM and IgD. So similar to the other section. So again, this axis applies to all these figures, by the way. So again, we have our double IgM up here, so we're getting that matching between IgG and IgM. Cells that are high in IgD, will be all up in the upper left, where cells highest in IgM only would be over here. So again, we talked about our B cells. Most B cells will express both of them if they're not been, they're kind of are mature naive B cells. So fluorescence activated cell sorting, or FACS, is a modification of flow cytometry to allow cell separation. Cells pass in droplets through laser beam And if they're tagged with fluorescent antibody, the detector activates electrodes, which have a charge given to the droplet, so a positive charge to the droplet with a labeled cell. So as we talked about, we had that laser coming down, and we had that figure with the 3 tubes. So once it detects that it's okay, it knows it has a yellow tag, it's going to immediately be sorted into this bin with all the other ones of the same dye. So here we can see it's sorting non-fluorescent cells and fluorescent cells into their own, using the charge, using the electrically charged plates to deflect fluorescent cells. And there can be different charges for different fluorescents, so we can move them. So yeah. These charged droplets are deflected by electrically charged plates and direct flows into separate streams. So again, this also gives us a way to collect them after and then do any further examination. So the identification of cells expressing certain cell surface molecules. So the use of antibodies specific for certain cell surface molecules to identify cell types of interest in a mixture of cells. This is what we're seeing in this figure. Fluorescently labeled monoclonal antibodies can be used to identify cells of interest. Using the process of fluorescent activated cell sorting, cells of a particular lineage can be physically separated from a complex mixture of different cells. So essentially similar to what we talked about earlier with fluorescence, but now in a flow cytometry, essentially we're sorting them. So here are positive cells, these will be sorted away, and these will be sorted in another way. So guys, I hope you enjoyed our last lecture on flow cytometry. Again, this is probably a more advanced one that you wouldn't see in most. Every lab, I know in my lab at school we don't have it because they're quite expensive and you need trained technicians. Like again, there's programs, 3-year programs dedicated to being a technician for these machines. So they're quite advanced, but a lot of, big school labs have these flow cytometry because they can do a lot more advanced studies. So if you go to a bigger school, they may have one. And I recommend getting a chance to look at the outputs and some of the images because it can be really, really cool. Like we talked about earlier, we saw those dyed pictures and it allows you to see some really cool stuff. So yeah, guys, I hope you enjoyed immunology and I'll see you in the next series of lectures.